Good afternoon students, welcome back to botany session. In these videos we are going to see nitrogen cycle and nitrogen metabolism. It is one of the topic of mineral nutrients. The chapter 11th chapter, mineral nutrients, subtopic is nitrogen cycle and nitrogen metabolism. What is called nitrogen cycle? What is the essential of nitrogen? Nitrogen, it is one of the important macronutrients essential for plant growth, that is used for the synthesis of amino acids, then proteins, then nucleic acids such as DNA and RNA. So for the synthesis of amino acids, protein is a building of the plant body, and nucleic acids such as DNA and RNA. For the synthesis of all these things, nitrogen is essential. So where is the source of nitrogen? Then what is called nitrogen cycle? It is a regular cycling of nitrogen among the living organism or in the reservoir pools in the lithosphere on the rock. It is a regular cycling among the living organism. These are called nitrogen cycles. Where is the source of nitrogen? Atmosphere is a source of nitrogen, but they exist in the gaseous state. So plant, they do not take in the form of gaseous state, it take in the form of ammonium ion, nitrite ion and nitrate ion. So for these conversions, certain microbes, free living microorganisms, they are actively involved for the conversions of atmospheric nitrogen into ammonium and nitrate to the soils. So the nitrogen cycle, it includes 5 stages. The first stage, fixation of atmospheric nitrogens. Atmospheric nitrogens. The second stage, nitrifications. Third stage, nitrate assimilations. Then fourth one is ammonification. Then fifth one is denitrifications. So these are the 5 stages that are involved in the nitrogen cycles. One is fixation of atmospheric nitrogen. Second one is nitrification. Third one is nitrate assimilation. Then fourth one is ammonification. Fifth one is denitrification. For easy, better to understand, I will draw the schematic representations. Atmospheric nitrogen. The atmospheric nitrogen that are existed in the gaseous state. Molecular nitrogen atoms that exist gaseous state. The plant, it is not a usable form for the uptake of by the plants. It is an unusable form. For the conversions of usable form, certain microorganisms are involved. So, by biological, how to fix the atmospheric nitrogen? Fixing of nitrogen is nothing but atmospheric nitrogen converted into ammonia. These are called nitrogen fixations. It is involved, for example, biological nitrogen fixation, industrial nitrogen fixations. Apart from that, electrical nitrogen fixations. So through this process, ammonium is formed. It is the first stable product, ammonium. The first stable product. The ammonium is converted into nitrite ions with the help of Nitrosomonas bacterium. This nitrite is converted into nitrate ion, NO3 minus nitrate ion by nitrobacter. By this process called nitrifications. And now the the ammonia is a first stable product formed during nitrogen fixation. Plant take in the form of ammonium ion, nitrite ion and nitrate ions. Among these, the plants are well adapted to take nitrate ion than that of the nitrate ammonia and urea. These process called nitrifications. So nitrification is nothing but conversions of ammonia into nitrate. Nitrification. Now the availability of nitrate in the soil, it is uptake by plants. Plant biomass. So now these nitrate ion utilized by plants for the synthesis of various amino acids, proteins, body builders proteins, nucleic acid synthesis. So from the plants absorbed by animal biomass. Because animal is the heterotrophic organism, they do not prepare their own food material, they derive the food from plant biomass. So both are, after death of both plants and animals, they get decay and decompose. Decaying with the help of a saprophytic organism such as bacteria and fungi. So after death of plants and animal remains, with the help of the saprophytic bacteria organisms, for example bacteria and fungi, they act on the dead bodies of plants and animal community, gets and decay and decompose to release ammonia by the process called ammonifications. So what is called ammonification? Conversions of nitrate into the ammonia is called ammonification. Conversions of ammonia into nitrate by the process called nitrification. So these are the overall scheme of nitrogen cycles. Now I will explain one by one nitrogen fixations. Fixation of nitrogen is nothing but conversions of atmospheric nitrogen into nitrate or ammonia. These are called nitrogen fixation. Which is actively involved in the nitrogen fixation. What are the certain stages, certain conditions are involved, essential for nitrogen fixations? Because conditions involve nitrogen fixation. The industrial nitrogen fixation, how the industrial, due to the presence of high temperatures, the union of nitrogen plus hydrogen to form nitrate. In the presence of high temperature in the industrial to form nitrate, it is a nitrogen fixations. Electrical nitrogen fixation, it is also called atmospheric nitrogen fixation due to the presence of lightning and thunder, UV rays, ultraviolet rays. The atmospheric nitrogen combines with the oxygen to form oxides of nitrogen, NO, oxides of nitrogen. Again, these oxides of nitrogen are combined with the water molecule to form nitric acids and nitrous acids. They make us acid rain. Through this process to form nitrate, it is also formed. The electrical nitrogen fixation and the industrial nitrogen fixations. So now the nitrate ions present in the soil, with the help of certain bacteria, they release, the process of conversions of nitrate into the atmospheric free nitrogen, come back to the free nitrogen by the process called denitrification. denitrifications. For example, Pseudomonas. Pseudomonas is a bacterium that involved in the denitrifications. Nothing but the process of removal of nitrate from the soil. Again come back to the atmospheric free nitrogen. These are called denitrification. So the industrial nitrogen fixation, electrical nitrogen fixation, it is the simplest way. It is a short process. But whereas in the biological nitrogen fixation, it is one of the long process. Certain bacteria, free living bacteria, and cyanobacteria, and rhizobium bacteria, Clostridium bacteria, Rhodospirillum, these are the free living bacteria that are actively involved in the fixation of atmospheric nitrogen into ammonia. It is one of the long process. So maximum the nitrogen fixations takes place in the leguminous plants. Leguminous plants are also called pulse varieties. Leguminous plants, for example, chickpea, Cicer aeratinum, Bengal gram, all the green gram, red gram, all the gram seeds, red gram, black gram, Bengal gram, all the pulse varieties, maximum nitrogen fixations takes place in the leguminous plants because of due to the presence of bacterium is called Rhizobium bacteria. See, it is a leguminous plants. So Rhizobium bacteria, it causes nodule formations. It is a symbiotic nitrogen fixing bacteria. They cause nodule formation in the root. These are the actual site for the nitrogen fixations. Actual site for the nitrogen fixation. If they fix the atmospheric nitrogen in the root nodules by these bacteria, certain conditions are essential. So what are the conditions are essential? The first condition, nitrogenous enzyme complex. It is essential. Nitrogenous enzyme complex is essential. Then second one is certain minerals essentials. For example, molybdenum, MO, then iron, then sulfur. So the molybdenum, it is a micronutrient that is essential for the formation of nitrogenous enzyme. It is a neat question. Molybdenum essential for the formations of nitrogenous enzyme. Iron, it is also essential, then sulfur also essential. Certain mineral nutrients essential for the fixation of atmospheric nitrogen. Then third one is ATP, energy is essential. The ATP is obtained from respiration process. What are the uses of ATP? The ATP which is used for transfer of hydrogen atom from the hydrogen donors such as glucose 6-phosphate, electron donors, glucose 6-phosphate, fructose 6-phosphate, sucrose, these are the hydrogen donor. So the ATP which is used to transfer of this hydrogen donor into the molecular nitrogens. So it is essential ATP. Apart from that, glucose 6-phosphate, these are the hydrogen donor. It is essentials. So these glucose 6-phosphates are obtained from photosynthesis. ATP obtained from respirations. Obtained from respiration. Then fifth one is anaerobic condition. Anaerobic conditions. Because anaerobic in the sense absence of oxygens. In aerobic condition, the enzyme, nitrogenous enzyme become inactive. The nitrogenous enzyme, it is highly sensitive to oxygens. In the presence of oxygen, definitely they become inactive. They could not fix the atmospheric nitrogens. So for that purpose, to create anaerobic condition, in the absence of oxygen, this bacterium, Rhizobium bacterium, along with these leguminous plants, to produce a pink coloring pigment is called leg hemoglobin. Leg hemoglobin, it is an MOFE proteins. molybdenum iron protein content. So the leg hemoglobin, it is a pink coloring pigment produced by combinations of rhizobium bacteria and leguminous plant. I mean the hemo part obtained from rhizobium bacteria. This globin from leguminous plants. So due to the presence of this leg hemoglobin to remove the oxygen, so it is called as a oxygen scavengers. It's nothing but removal of oxygen to make anaerobic condition that is suitable for nitrogen fixation. Remember, leg hemoglobin, it is a pink coloring pigment. It is highly responsible for the oxygen scavengers. Due to the presence of this leg hemoglobin, they remove the oxygen to make anaerobic condition. It is a suitable condition for nitrogen fixations. So the rhizobium bacteria, it appears in the pinkish color due to the presence of these leg hemoglobin pigments. So these are the fifth conditions. I will explain one by one. These are the conditions essential for nitrogen fixation. Nitrogenous enzymes, once again, minerals essential, ATP, glucose 6-phosphate, and anaerobic condition essential. So how fix the atmospheric nitrogen? The atmospheric nitrogen, it is a gaseous state. It is occurs in gaseous state. So these are substrate or dinitrogen molecule. Atmospheric nitrogen, it's a dihydrogen molecule atom. It's a gaseous state. There are triple bonds are present between the two nitrogen atoms. It is how to fix it with the help of nitrogenous enzymes. These are nitrogenous enzymes. So, for the gradual reduction of this triple bond by the addition of a pair of hydrogen, you know that's where the source of nitrogen atom, where the source, it is obtained from the glucose 6-phosphate. These are the sources of hydrogen atom. So this dinitrogen gets reduced by the additions of nitrogen, a pair of hydrogen atom, and finally these triple bonds are cleaved to produce 2 molecules of ammonia. These are nitrogen fixation. See, for example, these enzymes fix with the substrate. Nitrogen, triple bond, nitrogen, fix with the substrate to form combining of substrate. The next, by the additions of addition of hydrogen atom, these triple bonds gets reduced to form 2 bonds are present between the nitrogen molecules by the process called reduction. So here N2 H2s are present. Then further reduce by the addition of another pair of hydrogens. Another pair of hydrogens nitrogen. So these double bonds are reduced into single bond. So it further also gets reduced, the single bonds also further also gets reduced by the additions of another pair of hydrogen atom. 1, 2, 3, 1, 2, 3. So, and finally, these enzymes and the substrates, they gets cleaved or disassociated to produce ammonia. H, ammonia, H, hydrogen atoms. So there are finally 2 molecules of ammonia produced. The enzymes are gets disassociated. They are again going to fix the atmospheric nitrogens. If you make the overall equations, atmospheric nitrogen, here 8 electrons essential, then 8 protons also essential, plus 16 ATP is essential to produce 2 molecule of NH3 ammonia plus H2 hydrogen plus 16 ADP plus 16 inorganic phosphates are produced. So these are the overall equations of nitrogen fixations. Molecular nitrogen atom consume 8 electron, 8 proton, 16 ATP. The ATP is obtained from respiration to produce 2 molecule of ammonia, hydrogen plus 16 ADP and 16 inorganic phosphates are synthesized. These are the nitrogen fixations. The next steps, nitrification. So nitrogen fixation over. Conversions of atmospheric nitrogen into ammonia by the process called nitrogen fixation. A lot of organisms also that are actively involved in fixation of atmospheric nitrogen. The second stages of nitrogen cycle is nitrifications. Nitrification is nothing but conversions of ammonia into nitrate by the process called nitrifications. It includes 2 steps. In first steps, 2 molecule of ammonia is synthesized, combines with 3 molecule of oxygen to form 2 molecule of nitrate ion plus 2 molecule of protons, 2 proton plus 2 molecule of water is produced. This reaction is catalyzed by the enzyme is called nitrosomonas. It is an enzyme, nitrosomonas. These are first steps. In second steps, these 2 molecule of nitrate ions combines with a molecule of oxygen to form 2 molecule of NO3. It is a nitrate ions. This reaction is catalyzed by the enzyme called nitrobacter. So nitrification, it is nothing conversions of ammonia into nitrate by the process called nitrification. It is carried out by the bacterium is called nitrosomonas and nitrobacters. The second stage. Then what is the third stage? Nitrate assimilations. How these nitrates are gets assimilated for utilizations. First steps, nitrogen assimilation is nothing but nitrate 3. Nitrate is converted into ammonium ions. This process called nitrate assimilations. It includes 2 steps. In first steps, nitrate ion gets reduced into nitrite ions. Nitrate reduced into nitrite. Nitrite. This reaction is catalyzed by enzyme is called nitrate reductase. Nitrate reductase. But molybdenum, it is essential for the catalyzation of these enzymes. Molybdenum, it is a micronutrient that is essential for the formations of these nitrate reductase enzymes. First steps. In second steps, this nitrite ion further reduced into ammonium ions. This reaction is catalyzed by the enzyme is called nitrite reductase. Nitrite reductase. With the help of copper and iron is essentials. So the copper and iron, it is a nutrient or mineral nutrient that is essential for the formations of or activations of these nitrite reductase enzymes. So nitrate assimilation is nothing but nitrate is converted into ammonia in 2 steps. Then fourth one is ammonifications. Ammonifications. Then what is called ammonification? Nitrate, it is converted into ammonia by the process called ammonifications. Already we know that the nitrate NO3 which is present in the soil, that is a suitable form for absorption of plant for synthesis of amino acids and proteins. So now after death of plants and animals, they again decompose and converted into ammonia by the process called ammonification. We know that. So name the bacteria that are involved in the ammonification. Bacillus vulgaris. The other one is Bacillus ramosus. So these are the bacteria that are involved in the ammonification process. Then fifth step, denitrifications. What is called denitrifications? The process of conversions of nitrate from the soil again come back to the atmospheric free nitrogens. This process called denitrification. I mean the process of removal of nitrate or nitrogen from the nitrate into the atmosphere. These are called denitrifications. Nitrogen ions converted into nitrogens. Nitrate ions again converted into atmospheric nitrogen with the help of pseudomonas. Then thiobacillus denitrificants, thiobacillus denitrificants. Then Bacillus subtilis bacteria. These are involved in the denitrification. So nitrogen cycle over. It includes 5 stages. Next we are going to discuss nitrogen metabolisms. Then what do you mean by nitrogen metabolism? Already we know that ammonium is essential, suitable ammonium, nitrate, nitrate, it is a suitable form absorption of by the plants for the various synthesis of amino acids and protein synthesis. How these nitrate ammonia that is used for the synthesis of amino acid. Nitrogen metabolism is nothing but synthesis of amino acid. See, amino acids that contain amine group. Each and every amino acid that contain at least one carboxylic group, one or more amino group. So this nitrogen atom, nitrate, that is used for synthesis of amino acid by the process called nitrogen metabolisms. The first steps, ammonium assimilations. Ammonium assimilation. Then what is called ammonium assimilation? Ammonium ion converted into amino acids. Amino acids. These are called ammonium assimilation. It includes two steps. First steps, reductive amination. Reductive aminations. In reductive amination, the ammonium ion NH4 plus ammonium ion combines with the alpha-ketoglutarate acid to form the amino acids called glutamate or glutaric acids. Alpha-ketoglutarate acids with NH4 plus ammonium ions. Ammonium ion combined with alpha-ketoglutarate acid to form glutamate or glutaric acid. It is amino acids. Glutaric acids. It is an amino acids. This reaction is catalyzed by the enzyme is called glutamate synthetase. Glutamate synthetase. In the process of NADPH2, nicotinamide adenine dinucleotide hydrogen phosphate. Ammonium ions combines with the alpha-ketoglutarate acid in the presence of NADPH2 to form amino acids called glutamate or glutaric acids. During this reaction, one molecule of NADPH2 gets oxidized to form NADP plus. Oxidation as a result to form water molecules are formed. So first step, reductive aminations over. Then second one is transaminations. Transamination. Transaminations. Transamination is nothing but transfer of amino group of the one amino acid to the keto group of the keto acids. Because this glutamic acid is a main amino acids through which or from which there are 17 amino acids are synthesized. For example, transamination, example R1 H NH3 amino group COO. It is a carboxylic group, it is an amino group. These are the structure of glutamic acid amino acids. Plus another amino group C double bond O COO minus. So these are keto group. Keto group. So these are the amino group. Amino group. So transfer of amino group from one amino acid to the keto group of the keto acids. That's all. These are called transaminations. So through which the main group of main amino acids glutamate or glutaric acid. Glutaric acid is the main amino acid. Through this transamination, they can able to synthesize 17 amino acids are synthesized. So as a result, these R1 C double bond O COO minus. Next R2 C amino group NH3 C double bond O. So by transferring these, a lot of 17 amino acids are synthesized. The entire process of catalyzed by the enzyme is called transaminase enzymes. Along with the coenzyme is called pyridoxal phosphate. Pyridoxal phosphate. These two enzymes play an important role in the transamination. The pyridoxal, it is a derivative of vitamin called B6 vitamins. Pyridoxine, it's a B6 vitamins. It is derived. Pyridoxal phosphate, it's a derivative of B6 vitamins. Next, the third stage is of nitrogen metabolism. Catalytic aminations. First stage, reductive amination. The second stage, transamination. Third stage is catalytic amination. Catalytic amination is nothing but formation of amide. Amide that contain more nitrogen than that of the amino acids. So that is why the amide formations. The first steps, glutamate, amino acids, glutamates combines with ammonium ion to form glutamine. The glutamine, it is an amide. There are two amides are present in the plants. One is a glutamine, another one is asparagine. These are the two amides are present. Amide contain more nitrogen than that of the amino acids. The glutamine, it is synthesized with the help of enzyme is called glutamine synthetase. Similarly, asparagine, it is an amide synthesized with the help of enzyme is called asparagine synthetase enzymes. So these are the two amide are available in the plant groups. During this reaction, one molecule of ATP is utilized to form ADP, diphosphate. One molecule of ATP is utilized during this process. This reaction is catalyzed. The enzyme is called glutamine synthetase. Now, second steps, this glutamine combines with alpha ketoglutaric acid to form two molecules of glutamate, amino acid, glutamate. This reaction is catalyzed by the enzymes called GOGAT enzymes. During this process, one molecule of NADH2 gets oxidized to form NAD+. Then what is called GOGAT enzymes? I mean glutamine 2-oxoglutarate amino transferase. It is an enzyme. Once again, the formation of glutamine, it is an amine group, combines with alpha ketoglutaric acid to form two molecules of glutamate. This reaction is catalyzed by the enzyme is called glutamine 2-oxoglutarate amino transferase enzyme. During these reactions, one molecule of NADH, nicotinamide adenine dinucleotide hydrogen, gets oxidized to form NAD+. So this amide, how they are formed amide, we know that each and every amino acids, they contain one amino group and carboxylic group. For example, COOH, carboxylic group. These OH hydroxyl groups are replaced by amino group as result to form amide. Amide, it is the most essential for the plant because it transfer, it contain more ammonia, they easily transfers. Sorry, it contain more nitrogen than that of the amino acids. So thank you. Next videos we are going to discuss continuously. Next video. Thank you.